Hello everyone and welcome to today's LabTools webinar. I'm Catherine Lloydell, Scientific Technical Editor for The Scientist, and I'll be moderating our discussion. Today, our speakers, Dr. Shahin Mohammadi and Dr. Courtney Anderson, will be discussing the first single-cell transcriptomic atlas of the human retina and the validation of findings using RNAscope. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentation. To ask a question, simply click on the Ask a Question tab and type your query into the question box located on the bottom left of your screen. We'll try to address as many of these questions as we can during our Q&A session. Our webinar platform is user-friendly. You can expand the presentation window by simply clicking on the diametrically opposed arrows in the upper right-hand corner. This will maximize the display within your screen. The webinar will be archived on the Scientist website and will resend you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsor. ACD, a biotechnic brand, shortens the path to personalized medicine and enables research, drug development, and clinical applications by unlocking the power of RNA. ACD is a leader in the emerging field of molecular pathology, developing cell and tissue-based research and diagnostic tests for personalized medicine. Based in Silicon Valley, ACD's products and pharma assay services are based on its proprietary RNAscope technology, the first multiplex fluorescence and chromogenic in situ hybridization platform, capable of detecting and quantifying single molecules of RNA in situ. ACD has two product lines, namely RNAscope and BaseScope, consisting of assay reagent kits and 15,000 plus off-the-shelf probes, in addition to a pharma assay services business, which allows customers to run assays for their unique targets rapidly. Since its first launch in 2011, the technology boasts 1,200 plus citations, a new publication each day now, for single, duplex, and multiplex RNA analysis. Our sponsor has provided us with some helpful resources related to RNAscope, and we've posted these in our resource list located on the left side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Shahin Mohammadi. Shahin is currently a research scientist with a joint appointment at MIT Computer Science and Artificial Intelligence Laboratory and the Broad Institute of MIT and Harvard. His primary interest is to build robust, reproducible, and interpretable models that are inspired by, grounded in, and driven by core biological knowledge. He develops computational models and methods, mathematical formulations, and rigorous statistical techniques for problems at the intersection of computational, biological, and clinical sciences, including single-cell analysis. He has developed an extensive set of tools for analyzing single-cell transcriptome, epigenome, and multi-omics data modalities, which have been applied across a wide range of disorders, including age-related macular degeneration, AMD, Alzheimer's disease, schizophrenia and bipolar disorder, and spinal cord injury. In this talk, he will focus on age-related macular degeneration disorder and how using their newly developed framework, ACTIONet, allows us to identify a previously underappreciated association between a subset of glial cells, glial, and AMD, one of the leading causes of blindness in the elderly. Shahin?
Thank you very much, Catherine, and thank you everyone for attending this talk. So I'm going to quickly go over the basic of anatomy of the retina. So retina has a complex ecosystem of cell types that spans a bunch of different neuronal subtypes, including excitatory, inhibitory, photoreceptors, projection neurons of different types. It also has immune cells and other non-immune glial cells. So it is a miniature version of a whole, basically, brain system. So a lot of cell types that you have in the brain, you have analogous cell types of same origin in the retina as well. So it is used both to model brain, and also it has its own host of complex disorders, including AMD, that affects retina. So in order to better understand the complexity of the system, we studied it using two independent platforms, one droplet-based using 10x platform, and the other well-based using seqWell. And we independently, for different samples, we profiled them using either one of them and analyzed the differences in the two platforms and analyzed which cell types are consistently identified in both and what are the heterogeneity observed in each one of them. So I'm going to quickly go over the computational framework that we use in order to analyze them. The basic idea of it is that a lot of techniques that are used for single-cell analysis typically are single resolution, which means that they only consider one number of factors or number of clusters. And what we showed is that using the same number of factors, typically under or over fits to the some cell types, and it just is optimal with respect to a small number of cell types. So what we did is that we developed a multi-resolution technique that spans through a whole series of different resolutions and captures heterogeneity across all those resolutions in order to render a comprehensive picture of what's going on. And in a very crude analogy, it is like using microscopy and using a Z-stack in order to capture different views of the cell and then rendering them down to get a whole picture of the cell. The pipeline is, the whole framework is out both on GitHub, and the paper is on bioRxiv currently. But very quickly, it does use the previously defined method that we developed called ACTION, which is a decomposition-based technique in order to find a vocabulary or a language that can be used in order to represent and explain the behavior of the system. Using that vocabulary, then it tries to define the behavior of the system and defines a network of cell-cell relationships based on that. Using that network, we can use a host or different series of tools, including visualization techniques, clustering, coloring, and imputation that are all applicable to this framework. Then we applied the same analysis framework to the 10x data from Retina, and we, unlike previous methods that typically they first identify clusters and then annotate each cluster, ACTIONet framework is capable of individually annotate each individual cell and uses its local topology and local connectivity around the neighborhood of that cell in order to predict its type given a set of curated markers. So we spent, well, quite a while curating markers because a lot of these markers that we found in literature are not necessarily consistent when you look at the single cell data. We curated a set of markers that are consistently found to be active in both 10x and seqWell for all different major cell types of the retina, which is also available as part of the ACTIONet package among markers for other cell types. And you can automatically annotate each cell using the provided markers, which allows us to see that we capture majority of the major cell types. And we did a couple of rounds of filtering based on minimum number of expressed genes, as well as a method for doublet detection that allowed us to remove some of the cells that were likely to be of bad quality. And then in order to verify these annotations, we individually projected markers, known markers of different cell types, and we observed the different cell types that we expected that be the high locality of those markers. And then this is a very qualitative measure in order to quantify these numbers. We averaged the expression of the top marker genes, known marker genes, within cells that we marked as one cell type, and saw that the marker genes for each cell type is rather exclusively expressed there on average. And then we analyzed, using the same framework, the seqWell data, The 10x data I should have mentioned has 20,000 cells, and we annotated 9 major cell types. And with sequel data, we are looking at around 4,000 cells and similar cell types. And then we use the same framework to annotate and verify cell types that we identified. And then the next question that we wanted to answer or ask was, what are the frequencies of different cell types? And we observe a rather different frequency when it comes to cell types. RGCs are particularly different, and one spec that I would like to introduce that was not addressed in detail in the paper is the difference between the two platforms. And rods are typically larger than the size of a 10x channel. So depending on the size, if it goes horizontally versus vertically, there is a chance that rod cells break, and the RNA material from it will contaminate a lot of additional cells when we do single cell. And that is most visible in RGCs because it has the least amount of RNA quantities to begin with. And that was one of the sources of difference that we observed between the two platforms, which is explained here and also in the paper. And basically what we did is that we defined an average signature for each cell type, and we tried to match the differential profile of those signatures across platforms. As you can see, for majority, so the diagonal is basically matches, the correct matches each cell type from one platform to the other, so from sequel to microfluidics. And the diagonal dominant factor says that each cell type is mostly similar to itself. We can see that the least similarity is observed from RGC to RGC, and that is dominantly after, well, weeks of troubleshooting. We found that basically doing single cell for retina has the downside that the rod cells may break and may contaminate the rest of the cells, which in 10x we have observed that extensively, and we had to curate that data to remove all those contaminations. That being said, another interesting observation that we saw in the beginning was that there was a lot of heterogeneity that we didn't expect to begin with in molar glia. And we did build a sub-ACTIONet of molar glia, which is to say we extracted all cells marked as molar glia and did another round of batch correction and ran ACTIONet again. And we found 3 very distinct subtypes of molar glia, which we marked with FOS subgroup, called for A3 subgroup, and FTL subgroup. And those 3 subgroups, each one of them have additional markers, but interestingly, called for A3 is also known to be associated with AMD. First of all, we did validate overall with RNAscope technology the locality of and the localization of these markers in the cell, and basically their mutual exclusivity or shared localization, which you are going to hear a lot more later. I'm not going to go over details of that. And then we also asked about particularly called for A3 to see if there is any differences, regional differences between macula and periphery. And we observed that indeed there is a macular preference for expression of called for A3. And then we asked, given that this gene is related to the AMD, can we do a systematic analysis to see how different cell types, including molar glia, may play a role in onset or progression of AMD. And this is not a new thing. Glial cells have been long associated with the neurodegeneration in the brain. And it's not surprising to see that these glial cells also play a role in retina, which basically AMD is also a degenerative disorder, and it's one of the leading causes of blindness in elderly. And benefit of it is, from a data science point of view, is that there are a lot of high-quality GWAS studies that we were able to use. And we basically got all the variants, and we did different type of analysis. The first type of analysis that we did is that we looked at genes that were linked to those variants in the study, and there were two subsets. There were the leading ones that they were most likely to be causal, and then there was the whole set of associated genes, which was around 600, and the leading one was around 30. And then what we did is that for each cell type, we computed the specificity of expression of each gene in that cell type using Wilcoxon test. And then we asked, can we observe a cell type specificity for genes associated with AMD, or they are just non-specific? So if you look at panel B, for example, VTN doesn't show specificity in any of the cell types that we observe. That is to say, if you look at the distribution of that gene, expression of that gene, we are not able to associate that with the identity of cell types. Whereas other genes like C3, which is a microglia-associated gene, also we can see that it is expressed there. Interestingly, also we observe a lot of genes that are affiliated with molar glia, to basically a lot of genes that are AMD associated to be specifically expressed in molar glia. We picked 3 of those genes and basically used, again, RNAscope to verify them and see that indeed they have specificity and expression in the molar glia. And then we did a second round of analysis to basically, as an alternative view, we said, now if instead of getting those genes, if we look at set of all differentially expressed genes, we are going to have as input value of differential expression, and also we want to look at basically relationship between preferential expression of that gene and the genetic risk score that we get from the GWAS study. And we did 2 type of basically tests, alternative tests. One was, what if we use the same RNA-seq data, single-cell RNA-seq data, but for an alternative disorder, which here we use Alzheimer. The other was, what if we use a different RNA-seq profile, which is brain, but for AMD. And then also we did analysis for different disorders, for different traits as well, to make sure the association is correct. What we observed is that cones and molar glia and vasculature, both, like all 3 of them, are strongly in this study also, were associated with molar glia, which the 2 approaches that we took, the specificity and the risk prediction approach, provide an alternative views, and the fact that we observe molar glia to come up as top as both suggests that molar glia may have a role in AMD. And I'd like to use this opportunity to summarize the main contributions. First of all, we have the first single-cell atlas of human retina that also is profiled using 2 separate platforms, both SQL and 10x. We did some comparative analysis and found what are the cell types that are dominantly identified reproducibly using both frameworks, and the results are in the paper. And then we identified heterogeneity in molar glia and found 3 distinct subtypes of it. And we observed an association between differential expression of genes associated with molar glia and with AMD. And the data is deposited on the GEO database. The accession number is down there. And the code for ACTIONet is also accessible through GitHub. And with that, I would like to thank you all, and I will answer any questions you may have.
Great. Thank you, Shahin. As a reminder to our audience, you may ask questions using the question box at any time, and our panel will have a chance to address the questions after our next speaker. And that next speaker is Dr. Courtney Anderson. Courtney manages the Research Applications Group at ACD in Newark, California. For over the last 4 years, Courtney has been managing research projects in the ACD laboratories and with collaborators from around the globe to support novel scientific applications of the RNAscope technology. Prior to joining ACD, Dr. Anderson completed her postdoctoral studies in metabolic biology at the University of California, Berkeley. Dr. Anderson received her Ph.D. in molecular and developmental biology from the University of California, San Francisco, and her BA in human biology from Brown University. Dr. Anderson?
Great. Thank you very much for that introduction, and it's great to hear all this wonderful work coming out of the Retina Atlas. So today, I would like to talk about complex, highly heterogeneous tissues and how you can incorporate spatial analysis to interrogate these tissues. So complex tissues such as the brain and tumors, as well as processes and perturbations such as embryonic development, are comprised of multiple cell types and states. And precise transcriptomic characterization of these tissues can enable identification of new cell types, predictive biomarkers, new therapeutic strategies, and more. However, interrogation of these complex, heterogeneous tissues really requires a highly sensitive, specific, and multiplex spatial approach with single-cell resolution. Now, traditional transcriptomic analyses have relied on bulk tissue analyses or tools such as PCR and northern blot, which provide an average level of gene expression in a sample, but can lose the spatial and the single-cell information. And so this is akin to a fruit smoothie, which I'm showing on the screen, which can be a delicious drink, but you can only guess really at what is in that smoothie. And so with the advent of single-cell transcriptomic analyses, such as single-cell RNA sequencing, now they have been able to provide molecular information at the single-cell level. And to come back to the fruit analogy, can now provide you the menu and tell you what fruit and how much of each fruit went into that smoothie. However, these techniques still don't provide you with how those cells are organized in the tissue context, as tissue is a three-dimensional shape. And so this is where spatial analyses can come into play. Single-cell multiplexing spatial analyses that detect RNA can provide transcriptomic information with spatial organization while retaining that single-cell resolution. So now instead of an average blended smoothie with mystery ingredients, you can see all the fruit and how it is spatially arranged. And so one technology that can really assist this spatial analysis is the RNAscope in situ hybridization technology. It consists of 3 parts: a unique target probe that ACD designs against your sequence of interest, a signal amplification system that generates a very high signal-to-noise ratio, and lastly, visualization of individual RNA molecules as dots. This assay allows for the spatial mapping of almost all RNA species, including messenger RNA, long non-coding RNAs, splice variants, highly homologous sequences, and even point mutations in cells and intact tissues, all of which can be visualized with either fluorescent or chromogenic labels. And the assay can be performed on a wide variety of sample types, including FFPE tissues, fresh or fixed frozen tissues, and cultured cells. Now, the 2 key features of the technology are really its probe design and the signal amplification. These are all DNA oligonucleotide-based probes. We depict them as Zs to emphasize the fact that they have 2 regions linked by a spacer. The bottom of the Z will complement and hybridize to the target mRNA transcript, while the top of the Z is the base for the amplification tree. And so when 2 Zs hybridize in tandem to the target sequence, it creates a binding site upon which the preamplifier can bind and the amplification tree can be built. To each preamplifier, multiple amplifiers bind, and to each amplifier, multiple labeled probes will bind. And these labeled probes can have either a chromogenic enzyme or a fluorophore. And this signal amplification strategy yields very high sensitivity and allows for the visualization of a target RNA molecule as a dot. So every dot that you'll see in these images is an individual RNA molecule. And background is eliminated because the signal is dependent on the 2 Zs binding next to each other on the target sequence. If both Zs do not bind next to each other, then the preamplifier cannot form a stable hybridization and the tree does not get built. And so consequently, there's no amplification of non-specific hybridization. Now, a standard RNAscope probe will consist of approximately 20 of these double Z pairs that are all pooled together, and they're designed to hybridize next to each other along the target RNA. So you can imagine if you have 20 of these trees along your transcript, it really can allow for a tremendous amount of amplification and signal potential, and that's how we can visualize an RNA molecule as an individual dot. However, all you really need is a minimum of 3 of those 20 double Zs to bind in order for there to be enough signal generated for molecular detection. And so really taken together, the combination of the probe design and the signal amplification is how the assay ensures high sensitivity and specificity and generates a high signal-to-noise ratio. And so we actually offer 2 unique assays for the spatial mapping of gene expression. The first is RNAscope. This covers the majority of RNA species. This is approximately 20 double Z pairs in length, and it can target most messenger RNA and non-coding RNAs that are greater than 300 nucleotides. However, we also offer another assay called BaseScope. This assay, due to an additional amplification step, now allows for us to get down to as short as a 1 to 3 double Z pair probe. So that means now we can get down to as short of an RNA sequence as 50 nucleotides. And so this now opens up the possibility to look at RNA species such as splice variants, exon skipping, alternatively spliced variants. If you have any short or highly homologous sequences, such as MAGE family members or CDR3 sequences and T-cell receptors. You can even look at point mutations with this. It's very powerful. We can get down to that single nucleotide change in the tissue context. And this is just an overview of the entire portfolio. You can see the entire breadth of the products we offer. The assay comes in chromogenic assays, both brown, red, and a duplex assay. In addition, the multiplex fluorescence assays that we offer come in a variety of flavors. We offer them as a 3-plex, a 4-plex, and then also our latest high-plex assay, which I'll show in a couple of slides, where you can now detect up to 12 targets at the same time in the tissue. And then, as I mentioned, also our BaseScope assay, which is a chromogenic assay that can be a single-plex or duplex assay. And so what I'd really like to focus on now is how you can use this technology as it's related to neuroscience research. The majority of our customers really use the assay to target GPCRs. They tend to be notorious for having challenging antibodies to detect by IHC. And so an alternative is to turn to the RNA to identify the cells that are expressing these GPCRs. And similarly for ion channels as well. In addition, to visualize cellular activity in a cell type specific manner. So if someone is trying to look at if a neuron is active, you can look at immediate early genes at the transcript level, such as ARC or CFOS, see immediately when they come on and know when that cell is active and when the activity ends. In addition, and I'll show some examples, alternative splicing. The brain consists of the most splice variants more than any other tissue. And so with the BaseScope assay, we can now interrogate these splice variants, which contribute to the tremendous heterogeneity you see in the brain. And then lastly, and just as Shahin had mentioned and showed very nicely in his work, how you can confirm and spatially map single-cell RNA sequencing signatures in the tissue context. And so here are just some examples to show you how the assay has been applied. We can see very nicely visualization of specific cell types in the brain with various RNAscope probes marking the neurons, astrocytes, and microglia. And to hone in on some of those microglia, you can see visualization of specific immune cell types, both the M1 and the M2 microglia. And then these are some very nice examples looking at dopaminergic receptor expression. So DRD1 and DRD2, which are some of the most commonly used RNAscope probes that we offer. They very nicely and specifically discern the 2 different cell populations. In addition, cannabinoid receptors, so shown here is CNR1 in the mouse hippocampus, and also opioid receptors. We can discern the difference between both the mu, delta, and kappa receptors shown here. And then this is just an example, as I mentioned, of this marker CFOS. These are immediate early genes that are activated transiently and rapidly in response to a wide variety of cellular stimuli. And they're activated at the transcriptional level in the first round response to a stimuli before any new proteins are synthesized. So examining these genes at the transcript level is really critical to capturing their activity status. So RNAscope is ideal for this application. And so again, we're showing CFOS here in green in combination with the GPCR and cholinergic receptor CHRM3. And furthermore, we can also discern between ion channels as well. So we're showing the example here of the potassium channel, KCNJ3, and the sodium channel, ASIC1. Now, as I mentioned previously, the brain is one of the tissues that has the most alternatively spliced genes compared to any other tissue. This really helps to create an enormous amount of diversity in cell types. So from one DNA sequence, due to alternative splicing, you could have up to 5, 10, 20, even more splice variants just coming from one DNA sequence. And these can result in splice variants, isoforms, exon skipping, circular RNAs, and beyond. And this abnormal splicing is really implicated in neurodegeneration and also other neuronal disorders. And so as an example, I just wanted to show here circular RNAs. They're a fairly recently new class of non-coding RNAs identified where the 5' end of one exon will backsplice or join with the 3' end of another exon. This is a process that's called a head-to-tail splicing. They display tissue and developmental stage expression, but they're particularly enriched in the brain, and they also play a role in cancer as well. But because of their fairly recent discovery and really no other tools that can examine them in the tissue context, they don't really have a very clear or well-defined function yet. And so we were able to take advantage of the BaseScope assay now, and we can put probes on these unique exon junctions. So what we show here, there's a unique junction, basically the 7-9 exon junction for DL-GAP1. We can put a BaseScope probe on that will specifically detect the circular RNA. We can put a probe on exon 9, exon 10 junction. That will specifically detect the linear form. And then we can put a probe on exon 7, 8, which would detect both the linear and the circular. And you can see very nicely on the right-hand side of the screen, we can detect all forms of these RNAs in the hippocampus. Now, in addition, this is an example of a splice variant detection. This is a very nice study that came out of the NIH where they were looking at splice variants of ERBB4. The signaling pathway is known to be associated with the risk for schizophrenia, and ERBB4 transcript levels that comprise either the JMA or CYT1 exons are increased in the cortex of schizophrenic patients. And so it's really critically important to identify the cells that are expressing these distinct isoforms. And this is what we can see here. Transgenic mice were expressing GFP under cell type specific promoters for either GABAergic neurons, which are marked by GAD GFP, and then for precursor NG2 marker and mature or CNP oligodendrocytes. These were used to unambiguously determine the cell type specific isoform expression. And we can see that very nicely. So the JMA isoform is the major isoform in precursor and mature oligodendrocytes, whereas the JMB isoform is more prominent in GABAergic neurons. Okay. As I mentioned in that previous paper, they actually took advantage of a really unique workflow that we refer to as dual ISH-IHC or dual ISH-IF. This is a very nice methodology. Over 300 of the RNAscope publications use this methodology where they can look at RNA and protein simultaneously on the same slide in the tissue context. And so this is really a great way to help further characterize cellular gene expression. You can use this to also identify the origin of secreted proteins or visualize cell surface markers with an RNA of interest. And because of the similar workflows between the two assays, including sample fixation, pretreatment, hybridization, signal detection, and data analysis, you really can combine this now into one workflow. We do have some recommendations, though, to ensure success with this assay. First, they do all these dual ISH-IHC protocols do require some optimization. We recommend that you combine a working RNAscope protocol with a working IHC protocol, so making sure that your antibodies work well just when you do IHC alone. We also always advise to perform the ISH assay first, followed by IHC. And then also to optimize the antibody and protein detection with our pretreatment reagents. The RNAscope assay does have a protease step in order to access the RNA. That protease could degrade some endogenous protein, so you would want to confirm that your protein can survive that protease treatment and would still be detected by the antibody. And so this is just a nice example here showing how you can detect all the neurons that are marked by new N antibody by immunofluorescence. And then we use RNAscope to look at the gene expression for VGAT, VGLUT1, and VGLUT2. Now, and as very nice you saw in the earlier presentation, this has been a really a great field to start incorporating spatial analysis into single-cell sequencing workflows. Now, these transcriptomic single-cell studies utilize dissociated cells, and they can result in the loss of spatial organization of cell population in the tissue context. And they also can result in some bias or underreporting, as you can lose some cells during that dissociation phase. So validation and spatial mapping of these single-cell analyses can be obtained using an assay that retains spatial organization, such as RNAscope. And because of the multiplexing capabilities, as long as coupled with its high signal-to-noise ratio and single-cell resolution, you can now confirm high-throughput single-cell transcriptomic results and get that spatial component. And many researchers have been using this in their single-cell sequencing workflows in order to spatially map a cell atlas, visualize gene signatures of newly identified cell subtypes, classify highly heterogeneous cells, and even confirm publicly available data sets such as the TCGA and tabular mirrors. And currently we have over 140 publications that have used the RNAscope assay to validate and spatially map single-cell sequencing data. And so I just want to show a couple of examples of those publications. We can see here very nicely, this is a publication that came from Sten Linerson's lab at the Karolinska Institute. It was published last year, where they created a molecular survey of the mouse nervous system. They analyzed 19 regions from the central nervous system, peripheral nervous system, and the enteric nervous system, all by single-cell RNA sequencing. And this totaled more than 500,000 cells. And then they created a taxonomy of all those cell types that they identified, which is shown here on the bottom right. And so the RNAscope assay was used to validate the identity and spatial diversity of many cell types in this study. Here I'm just showing the distribution of astrocyte cell types. RNAscope validated the distribution of the neurotransmitters SLC6A9, which was abundant in the cerebellum, and SLC6A11, which was abundant in the olfactory bulb. In addition, ISLR and GDF10 marked the local astrocytes that were enriched in the olfactory bulb and cerebellum. And then last and most strikingly, the detection of MFGE8 and AGT in the tissue context by RNAscope revealed a distinct border separating the telencephalon, or the front of the brain, from the diencephalon, or the back of the brain. Okay. And then in this study, I wanted to highlight the ability to also confirm single nucleus staining or RNA sequencing data. Now, this is another method that can be used often in tissues like the brain, where it can be hard to dissociate at the single cell level. So researchers will dissociate single nuclei and study those, and they seem to do a very good job of recapitulating the entire cell gene expression signatures. So these were researchers at the NIH. They created an atlas of the adult mouse spinal cord subtypes, and they molecularly characterized 43 neuronal populations. RNAscope is used to confirm newly identified key genes for many clusters, including the cluster highlighted in blue, known as DI1, and then the cluster DE5, which is, DI1, which is marked in red, and DE5, which is marked in blue here. Furthermore, they then leveraged this approach to identify neuronal populations that were active following a sensory and a motor behavior. So RNAscope-ish was used for the validation of neuronal populations that were activated, which is indicated by the upregulation of CFOS expression in response to formalin-induced pain sensory behavior. And then they could map these back to the spinal cord and see where the activated neurons are in the spinal cord. Now, as I mentioned, furthermore, with single nuclei, this can also be used in human brain as well. This is a study coming out of the MGH and Broad Institute, where they created an atlas of the human Alzheimer's disease brain. Now, Alzheimer's disease is an age-related neurodegenerative disorder. It has high molecular and cellular complexity, which still remains to be poorly understood. So MIT researchers analyzed greater than 80,000 single nucleus transcriptomes from the prefrontal cortex of 48 human patients with varying degrees of Alzheimer's disease pathology. And so the power of this study is that it really provided a first blueprint for interrogating the molecular underpinnings and cellular basis of Alzheimer's disease. And so the RNAscope duplex assay confirmed cell type specific downregulation of differentially expressed genes. And this revealed significantly fewer excitatory neurons, which was marked by SLC17A7, shown here in red, with detectable NTNG1 expression, which is shown here in blue. And that was also reduced. This is again in patients from brain sections from patients that had Alzheimer's disease or no pathology. Now this is a very nice study looking at the atlas of vagal sensory neurons in the mouse. These types of neurons are necessary for the control of organ functions such as cardiopulmonary and gastrointestinal systems, but knowledge on the complexity of the neuron types is still missing. So researchers from the Karolinska presented a very comprehensive identification, classification, and validation of the neuron subtypes, both in the jugular and the nodose-derived vagal ganglia. And so I'm showing here the nodose ganglia. They also used RNAscope for the jugular ganglia. They used 25 RNAscope probes to not only validate the neuronal types identified, but also to quantify the relative proportions of these neurons. And this is just some representative data showing how they interrogated these neurons and could also get that single cell resolution of these genes, the gene signatures that they identified. And so now I would like to discuss our latest assay that we just released a couple months ago called the HiPlex assay. This is a very powerful new assay that now allows researchers to detect up to 12 mRNA targets in the tissue. It's very powerful, particularly for researchers that are working with single-cell RNA sequencing data. You can now confirm all those gene signatures with higher plexing in the tissue context and still retain single-cell resolution and single-molecule detection. The way that this assay works is that we basically call it 3 rounds of 4. There's an iterative fluorescent detection in where all 12 target probes will hybridize at the same time. The signal amplification tree gets built, as I mentioned earlier, so this is still based on the RNAscope technology I described at the beginning of my talk. However, when it comes to the labeled probe step, where the fluorophore labeled probe gets hybridized, you would only do the first 4. So you would do your targets 1 through 4, fluorophore labeling first. You would then take a picture of these four probes, and then you would cleave off just the fluorophore. This is just cleaving the fluorophore. It's not cleaving the probe off or stripping the probe or anything like that. So it's not harsh to the tissue. It has no effect on the RNA or the tissue morphology. We're just cleaving off that fluorophore. So after you cleave off the first fluorophores, you would then come in and add the next 4 fluorophore labeled probes, so targets 5 through 8. You would again take another picture from that region. And then if you were going on to do 12 targets, you could do stop at 8, or you can continue on to 12. You would repeat this process for the targets 9 through 12. Again, you would then have 3 images taken, and then we provide a precise image registration software that comes along with the HiPlex assay. This image registration software will stitch all of the images together, so you can now see all 12 targets in the same field of view at single cell resolution. Now this assay is compatible with fresh frozen and fixed frozen sample types, and you can really, I think it has really 2 great benefits in addition to just validating and getting more targets on your sample that you can validate, but also you can save precious samples by running more targets on fewer sections. If you have different combinations of genes you're interested in looking at, rather than having to do 3 or 4 sections, you can now do this all in 1 section. Furthermore, in tissues like the brain, where as you section through the tissue, you very quickly change structure within the brain, you can now visualize more cell types within the same plane of interest. And so these are just some examples of the data that you can get with the HiPlex assay. This comes from a customer who used 12 probes to examine the cortical layers in the mouse brain. And then we also had a recent presentation at the American Society of Human Genetics, where we presented this study where we validated the diverse striatal cell types that were identified in a publication that came out in 2016. They had analyzed over 1,200 single cell striatal cells, and they had identified all of these unique subtypes of the D1 and D2 neurons. However, they did not show any conformation or spatial mapping. And so these 2 subtypes, the D1 and D2 MSNs, were really characterized each by 8 target genes. So we thought to fully visualize these cell types, we could use our HiPlex 8 assay. And then in addition, if we use the HiPlex 12 assay, we could actually capture all of the MSNs in the same tissue context. And so this is just an example of that data here. You can see very nicely, this is characterizing now or visualizing the 8 genes that mark the D1 MSN subtype. And these are the 8 genes that mark the D2 MSN subtypes. And lastly, we can put all, as I said, detect all 12 targets that really capture all the D1 and D2 subtypes, and you can see all of those here on the screen in one image. Now, when you start getting into this higher plexing, it can be very challenging by eye to discern which cells are actually expressing all 12 of these targets, or even, more than 4 or 5 of these targets. So this is where the image registration software is really a nice tool. It allows for flexible target visualization. So here I'm just showing a screenshot of the interface of the software. What you can do is really toggle on and off which genes you want to look at. So you can look at just 1 or 2 genes, or you can look at all 12 genes, and you can identify which ones are actually expressing the combination of genes that you're interested in. And so, for example, that previous image I showed with all 12 genes, now we toggled off all the other genes and just focused on D1, D2. We can just see those 2 cell subtypes. And we can even now with this confirm that finding in the paper that showed D1 and D2, some cells can express both D1 and D2. And so in summary, I'd just like to show the adoption of the RNAscope technology. We had our first publication in 2011, and since then we have now had over 2,500 publications. And many of these are in top-tier journals. Over 25% of our publications are actually in some form of Cell, Nature, or Science. And furthermore, our top research areas are by far cancer and neuroscience. Again, so we are well adopted in these research fields. And so in summary, what I've showed you today is the RNAscope technology uses a very unique probe design strategy to dramatically improve signal-to-noise ratio and provides a strong specificity. We actually have over 22,000 probes already in our catalog, but if your probe is not, or your target of interest is not in one of those 22,000 catalog probes, we can easily design a new probe within 2 weeks for you. In addition, the high sensitivity of the patented amplification system really allows for visualization and detection of even low expressing targets. The multiplexing capabilities now with the newest assay, RNAscope HighPlex, we can detect up to 12 targets simultaneously in the tissue, but still retaining the single molecule and single cell resolution that RNAscope is known for. In addition, now with the new BaseScope assay, we also can detect more unique RNA targets that other technologies or other techniques can't even detect. We can detect splicing variants in short sequences now down to approximately 50 nucleotides. And then lastly, the power of these RNAscope and BaseScope assays can be complemented by protein-based assays such as IHC and IF for simultaneous RNA and protein detection on the same slide. And with that, I'd just like to thank you all for your attention. You can visit us at acdbio.com, or you can contact us at the email addresses on the screen. And if anyone will be attending the Cold Spring Harbor Single Cell Analyses meeting, we will be there with a poster and a flash talk later this week on Thursday. Thank you very much for your attention.
Okay, great. Thank you so much, Courtney. The audience has submitted several questions, so let's get straight to them. Shahin, this first question is for you. Did you analyze VEGF subtypes such as VEGF A, C, or D?
Sorry, would you repeat the question? What subtypes?
VEGF, VEGF subtypes such as VEGF A, C, or D?
No, we didn't.
Okay, no worries. This is another question for you, Shahin. This is about retinal pigment epithelium.
RPE,
Can you, RPE, can you clarify whether there is any RPE in the tissue analyzed for AMD samples?
Absolutely. We have actually had that request from a couple of readers already. We didn't include that in the version that we published, but yes, we have had it, and because of the focus of study and the small number, we separated that from the data we have. But anyone who is interested, we can share them separately with them. But yes, the sequel data specifically had more RP content.
Okay, great. Can you go into more detail about how the AMD enrichment score is calculated?
Sure. There were two separate scores. One, I cannot call it AMD enrichment. So where we started with the first analysis was a gene set, basically, a set of genes that came out of US study, and they were linked to the variants that they identified. So we started from those set of genes. And what we asked first was, is there any specificity, some type of specificity to any of those genes? That is to say, if I line up each one of those genes, let's say 600 genes, and I compute how specific that gene is for cell type A, B, C, and D, do I observe that that like one particular gene is very specific in one cell type, or maybe two, or maybe it's not specific to any? And then what we found is that a majority of these genes that are identified in the dual study where they were showing specificity for molecule. And that is to say, there is a significant difference in terms of average expression of those genes where we are comparing average expression of that versus any other cell type. That was number one. Number two was if we basically model one as a function of the other. So we use the MAGMA framework, and that's a standard type of GWAS analysis that is gene-based. And it does basically link between the risk scores and the preferential expression of the gene, and try to see if there is basically, in a very crude sense, is there any positive correlation between the risk scores and the preferential expression of the genes in that particular cell type. And in order to verify if our study is basically has both sensitivity and specificity, we tried an alternative tissue and an alternative trait to see if basically we observe like negative results, because it should be only positive for the tissue of interest, which is retina, and trait of interest, which is AMD. So if we are looking at AMD in brain, it should be negative, which was the case. And if we are looking at something completely different, trait that is irrespective of retina, in retina, it should be negative, which was also the case. And that would be a summary of it. Yeah.
Okay, great. Thank you so much. Courtney, this next question's for you. It's about the technology, the HiPlex assay. What fluorophores does the HiPlex assay use?
Yes, that's a great question. So the 4 fluorophores that come with the assay would be fluorescein, so that's the green channel, Alexa fluorophore 88. There's Psi3, which is orange, and that's Atto 550. Psi5, which comes in the far red, that's Atto 647N. And then for the 4th channel, it's Psi7, which is near infrared Alexa Fluor 750. And so if there are any customers that do not have the 750, you can modulate the assay a bit, where instead of doing 3 rounds of 4 targets at a time, or 4 fluorophores, you would do 4 rounds of 3 fluorophores.
Okay, thank you. Courtney, this question is also for you. Can dual ISH-IHC be performed on an automated stainer?
Yes, I didn't touch upon that. Actually, almost all of the RNAscope assays can be performed on an automated platform. We support them on the Leica Biosystems Bond and the Ventana Discovery instrumentation. And in addition to with the dual ISH-IHC, yes, that specific methodology can be performed on either the Leica Bond or Ventana Discovery platform.
Okay, great. And this is another question about RNAscope. What neuroscience-related probes are available for use with RNAscope?
Yes, so we have quite a few probes, or at least as I mentioned, we have over 22,000 in catalog, and neuroscience is one of our top research fields. So there are many neuro-related probes. I would say the most popular probes are typically for GPCRs, such as DRD1 and DRD2, since they are notorious for having poor IHC antibody specificity. But we also offer cell marker probes, such as RBFOX3 and GFAP, S100B, AIF1, also markers for the glutamatergic and GABAergic neurons, which I showed here, vGlut1, vGlut2, and VGAT. But, again, if if you don't find a probe in the catalog, ACD can design a probe against any target. All you have to do is just submit your sequence if it's a new sequence you've identified, or if you know the NCBI accession number, you can just send that to our probe design team, and they will work with you to design a probe that's specific for your target.
Okay, great, thank you. And then I have another RNAscope question here. How long does the RNAscope assay take to perform?
Yes, this really is meant to streamline in situ hybridization assays. So, traditionally, the whole thing within traditional in situ could take, at least 4 days just for the assay, up to, 3 weeks if you're counting making a probe. So now, between our probe and just the assay itself, the RNAscope can only take about 6 to 8 hours, depending on which assay you're doing. So it really is meant to be a one-day assay, and one day you can get your data. So that, like, for instance, the manual chromogenic assay takes about 8 hours. You could even divide it up over 2 days if you want. The fluorescence assay, so the 3-plex multiplex assay I mentioned, that one is our shortest assay. It only takes 6 hours. And then some assays, such as like the methodology, the dual ISH-IHC, that one can take longer since you have the addition of protein staining. So that can be anywhere between 11 to 14 hours. And then also if you do any of the automated assays, while they may take about 12 hours on the instrumentation, they actually only require 30 minutes of hands-on time. So it really is a fast assay for researchers.
Okay, great. Shahin, this is another question for you. For those of us interested in leveraging the Retina Atlas data in our disease cohort, how can we maximize the data set and eliminate batch effects between your control data set and our disease cohort?
Sure. Well, batch correction is obviously a matter of continuous debate. There are two platforms. One is Harmony, and the other is Mutual Nearest Neighbor. And obviously, there is the basically Seurat version of it as well. In the ACTIONet framework itself, we incorporated both Mutual Nearest Neighbor and Harmony. So if you have, let's say, data of your interest, you can just combine it with the Retina Atlas and add a vector that says which data set it comes from. And you pass that vector to the function, it automatically corrects the batch effect for you. So it is already documented in the ACTIONet framework, and you can easily batch correct and incorporate it in your data as well. And I have to emphasize the data here is, while we studied AMD, comes from healthy retina, not from disease. Like, all the samples were healthy samples, just to make sure.
Okay, great. Thank you for that. Courtney, we have another question about their assay. What is the difference between the V1 and V2 fluorescent assays?
Yes, so the V1 assay is up to 3 targets, and it's really designed to work with fresh frozen or fixed frozen tissues, whereas the V2 assay, you can detect up to 4 targets, and it's primarily meant for FFPE tissues. It basically uses TSA opal dyes and fluorophores so that it reduces the autofluorescence that you can typically get from FFPE tissues.
Okay, thank you. And we have time for just one more question. Courtney, this one's for you. Can you detect multiple targets at once with BaseScope?
Yes, you can. We offer a duplex assay, so you can actually detect up to 2 targets with the BaseScope duplex assay. So you can look at, your splice variant or exon junction, and then you can also combine it with a cell marker, so you can get cell type specificity. In addition, some researchers also combine BaseScope with IHC, so you can actually get a protein target detected. So there are 2 methods of getting multiple targets with BaseScope.
Okay, thank you so much. Unfortunately, that's all we have time for today. If you have any further questions or would like clarification on anything, please consider reaching out to our speakers directly. Their email addresses are shown on the screen right now. As a reminder, the webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Shahin Mohammadi and Courtney Anderson, as well as our webinar sponsor, ACD Bio. Thanks everyone, and goodbye.